All right, that was great. So, all right, so next up is Nathalie Agar from Harvard Hospitals, and she's going to talk about mapping drug distribution.
Thank you, Forrest. And I'll spare you the coughing spells by being able to move. Hopefully, I'll use this microphone, but remind me if I get out of alignment with the microphone. So today I'll talk to you about the work we're doing on mapping drug distribution and response in GBM. And I'd like to say, to get started with, is that we don't have a unifying theory yet. We still have 4 years and 3 months to come up with one. But that basically we'd like to eventually, as Forrest said, well, I always feel bad when I put this together. I'm like, okay, I'm just showing tools and results, and we don't have an answer yet. But we're just started. So today I'll show you some of the technologies that we're bringing together into the center to be able to look at drug distribution and correlate to response. I do have to make this disclosure per our institution. So the imaging platforms that we've been developing for drug development and their use is to be able to look at distribution of drug and their metabolites without any molecular labeling. So we don't want to change the chemical entity by being able to image them. The second one is to be able to image and quantify drug delivery through the blood-brain barrier, and I think by the end of today you'll be completely brainwashed about the importance of the blood-brain barrier, but that's how we all came together, because we think it's important. And the initial work that we've done in this field is to look at preclinical animal models, but we've already moved those technologies into helping clinical trials. And ultimately, what we want to be able to do is to do an integration, a spatial integration of pharmacokinetics and pharmacodynamics. So one of the main tools in the center that we've been using to be able to do this is to use matrix-assisted laser desorption ionization mass spectrometry imaging, MALDI MSI. So in this case, what we do is we take a frozen tissue section, we prepare it with a small organic acid onto the surface that will serve to transfer the energy coming from a radiant laser to the analytes into the tissue so they can desorb and ionize and go into the mass spectrometer. So if we raster our laser in an XY fashion over the tissue section, we generate a full spectrum of a mass range of interest, forming pixel that can, and then the data can be mined to be able to look at the distribution of molecule green, molecule purple, or molecule yellow. So taking this approach, we can map the distribution of hundreds and sometimes thousands of molecules in the tissue without requiring any molecular label. And the reason we're able to do that is we use high resolution or high mass accuracy mass spectrometry. We use well, here this one was done with a 12 Tesla MALDI-FTICR instrument. We typically use a 9.4 Tesla magnet. But what we are looking for is, for example, if you have Erlotinib here, you have the chemical formula. We're looking for the monoisotopic peak, and then the isotopic distribution of the, considering the natural isotope of the different elements into the formula. But also at this, with this type of instrument, we can even look into the isotopic fine structure. So here, if you look at the 2-carbon-13 peak right here, if you zoom in, you can look at the contribution of 2-carbon-13 versus 1-oxygen-18 versus a carbon-13 and nitrogen-15. So it's this type of mass accuracy and resolution that allows us to look at the distribution of drugs into very complex environments such as tissue. So this is very old data. It feels like a kindergarten drawing that I took back home, but this illustrates the point. So in this case, we were looking at Erlotinib as basically the first drug we ever tested doing this. And in the liver, we can image the distribution of the drug, but also some of the different metabolites of Erlotinib. And in the brain with the tumor, we didn't have the nice models from Jan at that time. This is a U87 model. We can look at the distribution again of the drug and also some of the pharmacologically active metabolite. So using that technology, we were able to look at 10 over, I think, about 15 metabolites of Erlotinib into the different organ, including the pharmacologically active metabolite. So as Jen mentioned, our interest is in looking into the blood-brain barrier, and what we want to know is whether the drug is found within the lumen of a vessel, or is it able to reach the cells into the tumor parenchyma or infiltrated brain parenchyma. So to do that, what we've done is to use heme, which is a cofactor of hemoglobin found in red blood cell, as a marker of the vasculature. And we validated this by doing fluorescent injection into in an animal, which is an accepted marker of the vasculature, taken serial section of the tissue, did microscopy, fluorescence imaging, then did covered with the matrix, did mass spec imaging of heme, of the fluorescent, and then you have a very strong correlation of those images. So now what we can do is to use heme as a marker of the vasculature and see where the drug maps in relation to that marker. So as a positive control, we turned to PI3 kinase inhibitor, BKM120, which had been accepted to be very blood-brain barrier penetrant, and this is an overlay here and shows basically when a drug is permeable, we see a homogeneous distribution like this. And this was at 4 hours. But when we laid out the different animals that we had done, you have here all the animals at 4 hours. We didn't see the same pattern in those other animals. But if you look at and we could see drug penetrance here at 2 hours. And what we realized is that those were not all sectioned in the same plane. So when the animals didn't have a tumor, the tissue section were taken very frontally, very quickly into the brain. But when the animals had a tumor, we went a little further and went into the plane of the lateral ventricle here. So what we did is we zoomed in onto this animal and did sectioning from the frontal to the posterior part of the brain and saw that we had accumulation in 2 different planes where you have the opening of the lateral ventricle and closer to the cerebellum. And here, I'm assuming you see some green here, but I can't. All I see is glare, but usually there is green here. And basically, we have known that the blood-brain barrier is not the same throughout the brain, that there are differences in different regions, which is another factor that we're not taking into account when it comes time to treat patients, that where their tumor is located might be more or less accessible by drugs. It hasn't been a problem because none of the drugs make it at all. But hopefully this is something that we can start considering. So you've seen already some of those graphs. This was a negative control that we turned to, RAF265, a BRAF inhibitor that works really well on sub-Q implants, but when we put those tumors into the brain again, we lose the efficacy. So what we thought we would see doing the imaging on this animal is no drug accumulating at all. And so this is the animal with a tumor here, a non-tumor, and a vehicle animal. But instead, this is what we saw. We saw a strong accumulation of the drug into the tumor of the animal, but with practically no response. So these images were acquired at 125 micron spatial resolution. So we went back to some tissue sections and acquired images at 25 micron. And what we can see when we do the co-localization of the drug and the heme, most of the drug is found within the vessel of the tumor. So what we're seeing now is a spectrum of drug distribution that seem to be affected by both perfusion and diffusion. And if you look at the CD31 stain of this animal, you see those bloated vessels. Some are dead-ended. So if you have a rush of drug that will be going there, it might just be accumulating in here without being able to go across the vessels. And Jen showed you some of the results here, but what I wanted to show, again, to emphasize the fact that contrast distribution does not mean drug distribution, and I have colleagues who study the blood-brain barrier and use the contrast as a marker of distribution, is the study here where we overexpressed VEGFA, and you have almost a match here for the mass of the drug that we were looking at with the Gadavis, the contrast agent into the tissue, and we can see very different distribution from the contrast and the drug, showing that basically there's more to it than just the mass of the molecule. So our group came together, and this is busy, you're not meant to read everything to this, but this is, I'll take you through one of the studies that we're getting ready to publish on using Erlotinib as a model. So what we created here are animals, and I say we, this is all done in Jan's group, all the animals. We created a series of animals with flank that were treated with 3 doses over multiple days, vehicle, and then we did at different concentrations. So the idea is that in the drug, invariably, you'll have different drug concentration. You might be treating at 100 mg per kilo, but not so much will be going into the tumor. So and we don't have a response, but Forrest's group has been showing that the tumors develop resistance and adaptation mechanism, and I won't go into the details because I would sabotage it. But that basically what we wanted to look at here is that could we see differences in response at different concentrations. So we worked from the flank tumors to be able to get some molecular information intracranial. And then we also did a series of animal with just 1 single dose for 2 hours to be able to acquire some of the spatial information on those animals and see if we can make a correlation between the response here to mapping into the tissue. And then I'll take you into detail. So basically now what we want to be able to do is to acquire multiparametric data under those drug treatment condition and see if we can eventually come up with this model where we would be able to predict drug distribution and response from some basically non-invasive imaging such as MR or maybe PET or a combination of MR sequences. So what we did, some of those animals that I showed you, actually some were highlighted in red, were imaged by MR prior to sacrificing. So here is one of those animals, and each MR plane is 200 micron. So between 2 MR plane here, we can do all of those analyses, and then I'll take you through some of those so that we can provide characterization of drug distribution, tissue characteristic, and response. So in the first part that I'll be talking about is to look at the drug analysis as it relates to H&E stain and look at the distribution of some biomarkers with mass spectrometry imaging. So 1 animal per, and I might make it look a little easy here. They're not exactly just pictures. So 1 data set here when we want to do a 3D reconstruction where we take sections from the frontal to the posterior part of the brain. Each tissue section here is 70 gigabyte of data. And 1 full reconstruction like this can be 2 terabytes. So needless to say, we don't do this on every single animal that Jan produces. But basically, we try to extract some of the information from here. So here you can look at the distribution of Erlotinib throughout the brain in the pharmacologically active metabolite M13, M14. But we can also tap into some of that data, some of those spectra, to look at some biomarkers. So in this case, we have what appears to be a lipid here that maps very well with the tumor, and use another biomarker that maps with the normal brain. And what we do is do a 3D reconstruction where we can look at the distribution of the vasculature throughout the brain, of the lipid, of the drug, and of the metabolite. And even though these might look to be very similar, I just put the spectra here. There's 10 times more Erlotinib than there is metabolite, but it seems like that metabolite is distributing a little more broadly than the drug itself. So maybe it would be a strategy to work with that metabolite rather than the drug itself. So all of those images, we can look at them in a qualitative manner, but what we've been working to do now is to develop more quantitative tools so that we can bring all of this together. So in this case, we extract co-localization index, where now we're looking at co-localization between the heme, the tumor biomarker, drug metabolite, and the drug. And basically, almost as a control here is that the tumor biomarker and the blood are pretty much mutually exclusive. And what co-localizes the most, basically the higher the number, the higher the co-localization, is the drug metabolite with the parent drug, which is also making sense. But we can see differences of distribution between the drug metabolite and the parent drug as they relate to the tumor biomarker. And we can see, we saw in the previous, and I emphasized on that the metabolite seems to distribute a little more broadly. So I like to say that we work almost under peer pressure, is every time we come up with a result, then a colleague says, but those are nice pictures, but how much is there? So then we had to develop the approach to be able to be quantitative. And when I started doing MALDI mass spec and MALDI mass spec imaging in particular, that is not even 10 years ago. There's no way we thought we could ever be quantitative. And actually today we are, and it is quite reliable. So what we do in this case is we prepare a tissue mimetic model. So we prepare tissue extract that we spike with known concentration of the drug that we bring into a gelatin mold. And every time we put a tissue section on the glass slide, we take a section of that of that tissue mimetic so that we can calibrate our signal. We generate a standard curve and just calibrate the signal. But basically now we can look at the drug content in the overall brain here segmented with the red outline, but we can also extract how much drug there is just in the tumor itself here, and you can see that those numbers are quite different. But we can go into more details, and I don't know, you mentioned heterogeneity. I think you meant molecular heterogeneity, but we can also look into spatial heterogeneity, and we can further dissect out the tumor here and see that are different drug concentration in different parts of the tumor. And so then we applied this to look at basically what are we doing when we're comparing a flank animal with intracranial tumor. And we can never achieve, basically, those are the three brains treated with 33 mg and 100 mg per kilo versus the flank, and we're never able to reach what we see into the flank at 100 mg into the intracranial tumor. So another component that we brought to our Physical Science Oncology Center grant that basically gave us the title of physical is to use SRS microscopy. And in this case, we use stimulated Raman scattering microscopy imaging to look at tissue architecture, again, being label-free. So what we're looking at is the vibrational state of molecules, and in this case, we can't be as specific as with mass spec. We're looking at the CH2 vibration mostly found into lipids versus CH3 in proteins. We also use on that two-photon system an absorption mode where we can look at heme. And what it allows us to do is to look at the cancer cells that are in blue as they invade here into the normal brain. And I just love this picture here. Basically, you have the donut-shaped red blood cells that you can see in tissue without any molecular labeling. We can also use second harmonic generation where we look at collagen networks. And basically, this is what one of those tissue section looks from that Erlotinib study, where you can see the more lipid structure of the brain. And you can look into the tumor here, which is mostly dense cancer cells, some of the main vessels here that we can also see. And if we zoom in some of those regions, this was just an optical image, we can overlay basically the tumor delineation with normal brain. And as it relates to H&E, we've developed software where we can co-register those images and being able to validate what we're seeing with the SRS. And the idea now is that I mentioned that mass of a molecule is one of the important physicochemical property of a molecule that will dictate whether it penetrates the blood-brain barrier or not, but lipophilicity is also one of them. And if we look at the effect of those lipid structures here as they relate to the tumor and into the brain, I think we do need to start taking this into consideration. And as far as I know, there's no very good immunohistochemistry way of doing this. So what we are doing is correlating, and I say we, that's when that unifying theory will come together, is basically looking at the tissue characteristic by microscopy, by H&E, and correlate this to, in this case, this is drug and one of the biomarker, but also drug distribution, and then the list goes on to bring them together. And now, basically, that was pharmacokinetics. Now, what we want to look into more is pharmacodynamics, basically the drug response. So in this case, even though we have a lot of information in those spectra, the proteins are very difficult to image and not as specific, not as high resolution. So immunohistochemistry is still the best way to go. So on those tissue section, we can use simply immunohistochemistry and look at tumor response to correspond. So in this case, here we see that EGFR was indeed affected. And now Forrest's group has been doing phosphoproteomics, looked at over 210 tyrosine phosphorylation sites. And again, I'll let him speak to the more details of this. But basically now comparing the flanks of those animals that were basically placebo and the ultra-low dose and different drug concentration here, we can see the EGFR network being inhibited, but also starting to see some other pathways that show adaptation. And in that case, there was more heterogeneity into the adaptation and resistance mechanism pathways. And here, so those experiments we can only do so far using the flank. So we don't have the spatial distribution, if you want, into the brain tumors. So what we are doing, and this is with Jan Tran's group, is to take some of those sections and do laser capture microdissection, where we basically, in this case, divided the tumor into 4 quadrants and do mRNA sequencing to look at the tumor response. And we are supposed to see more of those results this afternoon as we're getting all together to put that paper together. But I think at a glance, initially, some of those results correlate well with the phosphoproteomics. Am I correct? Yeah. And now, basically, the big task is to be able to bring all of this together. And one of the limitations in bringing the imaging data together is that every time we take a different tissue section and subject it to a different treatment, we introduce tissue like distortion into the tissue, making it very challenging to delineate tumor and incorporate response into this. So basically, this is really fresh from a few days ago, work that Elizabeth did, starting to develop some data analysis workflow to look at if we would be able to develop some pharmacodynamic approaches from that mass spec imaging data that we are already acquiring. So in this case, from a very small mass range that had been used to image all of those, we do this whole pipeline of segmentation, spectral comparison, to be able to eventually identify biomarkers. But in this case, Elizabeth was able to do peak picking for, and I can't see it now, but 338 peaks that we are working from. So now if we, and some of the questions we have amongst ourselves and that we've received criticism for is how does the flank replicate the tumor that we are seeing into the brain? And here, if we do some segmentation and use 2 different clusters, basically at the molecular level, those tumors into the brain look very much like the flank tumors, which is, I think, a level of good news. But if we start subdividing some of those clusters here, we are starting to pick up some of the tumor heterogeneity that I think we could start using to be able to look at and correlate to drug distribution. So one of the first molecule of interest, if you want, if we look at here the drug distribution, this is the same layout that I presented before. There is this one, and this is so hard because I can't see it, but basically here, when we have no treatment, there is this molecule at M/Z 448 that accumulates very strongly into the tumor, but into the tumors that are exposed to the treatment where drug accumulates, we actually see a loss of that molecule. And if we zoom in here, we can see that the drug is actually the marker is just at the outside of the tumor here when the drug is really highly concentrated into the tumor. So we went back into the 3D data, which was acquired differently, and in this case we have multiple plane, and we see the drug distribution, and again, we see even this rim forming around the tumor that seems like we might have something of interest. So because we're using a high resolution instrument, high accuracy, based on the mass itself, it seems like that molecule could be one of those 2 here but that we will be looking more into it to see if this one or any other one that we could pick up could start being a bridge between the tumor response and what we're seeing with the phosphoproteomics and mRNA sequencing. So basically not just looking at the target itself, but seeing some responses when the cells start responding to the drug. So in terms, if we take all of this into consideration for clinical trials, it's only a few years ago that we started being able to give drugs to the patients prior to surgery, take a sample during surgery and send this to LC-MS analysis to see if the drug makes it to the tumor. And again, the criteria that we've had is basically asking the surgeon, can you pick in contrast versus non-enhancing region? But what we have done right now is, in a number of trials, this is one example here, that drug that I presented early on that is very permeable, BKM120, this is an MRI from a patient at the time of first surgery. Patient recurred 8 months later, received 8 days of treatment, 100 mg daily. And at the time of surgery, we took 2 samples from the enhancing region here and digitally registered on the preoperative MRI using neuronavigation where the drug came from. And more what we're doing is trying to map the drug distribution into the brain this way to see how it relates to the 3D anatomy of the tumor, but also as it relates to the 3D anatomy of the brain if we're taking into consideration the fact that the blood-brain barrier is not the same throughout the brain. So, and when we did the detailed analysis of one of those 2 samples here, this is a delineation of the necrotic region of the tissue. This is where most of the tumor is, and this is basically normal infiltrative brain. And this is where the drug went. So we thought this was going to be a home run. It was a very permeable drug. The patients have not been responding, and this is not the answer. At first, we thought maybe that is a little bit of it, but the drug turns out to be not as specific. But just to say that historically, we would just fail in the trial and put it away and move on to the other trial. In fact, in the IPG, I think we've been running 250 of those trials over the past 30 years and don't learn much from them. So now what we can do is to do this type of characterization. Then we can also do the quantitation. This is all of the patients from one of the cohort where we were able to do the quantitation and compare to the LC-MS analysis from a CRO. But more importantly is that now what we can do is to take, for example, this sample here, which had 800 nanogram per gram in total, but to start delineating 2 different regions into the tissue and be able to correlate with histopathology and hopefully with drug response. So I alluded to this. This is a typical spectrum from 1 pixel of those images. The drug signal is this teeny tiny peak here that we have in the full spectrum. This is the heme peak, and all of those hundreds, if not thousands of peaks here are waiting to be tapped into as hopefully biomarkers. So we tried to do this very qualitatively on this first trial. So this is a sample from the patient at the time of the first surgery who was not treated with any drug, and we can look at the distributions of different, what appears to be mostly lipids in this case, and compared to the patient once treated with the drug, and we can see some striking differences with some of those markers. And the story is not that simple. There's been 8 months in between and other treatments, but basically now and I told you that I was not going to tell you the whole answer to everything, but rather that we've been putting tools together that hopefully can help us provide more information in terms of where do those drugs go and how do they affect the tumor so that hopefully we can develop smarter drugs. So in summary, I've shown you some of the platform that we've developed into the center or part of the platform to correlate quantitative drug distribution with tissue characteristics and then an integration of drug distribution with drug response studies, and that we can apply those all the way from preclinical animal models all the way to clinical trials. And in fact, some of the early work that we did on MK1775 that Jan showed earlier, we received an award from the NIH to study the clinical trial now from those patients using all of our combined technology and expertise. And this is one of the trials that we're studying. There are a few other ones. So this is extremely collaborative work, and I'm just fortunate to be here to talk to you about this. And this is the team where we do a lot of this work into the operating room here, but I did not touch on that. And this was an answer to my postdoctoral advisor saying that this was not an acceptable slide because it's too small. So thank you very much. Yes?
Is an implication of your work that clinical trials on the effectiveness of drugs for these tumors should be stratified by the location of the tumor in relationship to the cerebral vasculature?
I think we're far from there, but I do think, yes, eventually we should take this into consideration. And the neuro-oncologists we work with, when they see this, they got discouraged at first, saying, well, there's another thing, but that they had never considered. But yes, I do think that we should take that into consideration. I think we've been fancy in trying to develop targeted therapy that are very specific for one molecule, but we are not taking into consideration any subtleties in pharmacokinetics, and it's extremely important when it comes to the brain. So.
Thank you. This being the Koch Institute, let me ask an engineering question. Neurosurgeons will do so-called craniectomies, which means take off the skull, half of the skull in patients who have strokes or traumatic brain injury, to allow the brain to swell. So I have a hydrostatic question, and this is for any of the speakers or anyone in the audience. Has anyone looked at these animal studies doing a so-called craniectomy, taking off the skull, half of the skull, and seeing whether drug delivery is enhanced that way? Because when you look at these flank tumors, clearly they can expand and contract, but in the skull, they can't. They're fixed.
We haven't, but there are definitely a lot of different things that we'd like to look into. There are factors, again, we're just looking at the region and the chemistry of the compound, but there are also factors even that would mimic that, such as the circadian rhythm would also have a different permeability throughout the day, and then this is another factor. We're also using focused ultrasound to transiently open the blood-brain barrier. Jen has more.
Just kind of a little bit to your point, presumably, I think the idea from a craniectomy is to decompress the brain so that there's less pressure in the context of pathology, and certainly we see increased intracranial pressure in patients. In the mice, when we're starting treatment, the tumors are relatively small, and they're happy as anything. So I think kind of they don't exhibit the clinical signs of increased intracranial pressure, at least. And mice are very sensitive, so that once they start exhibiting signs of intracranial pressure, they're dead in about 48 hours in these tumors. So there's a fairly narrow window. So I think it's an interesting question, because I think the question really is intracranial pressure limiting drug delivery, which is a very relevant question, but I don't think the mouse model would be a good one to test that way.
Bill, that's something to add?
Just briefly, one of the issues, and Nat alluded to this, is when you do that, then you're starting to change some of the glymphatic flow, which is kind of a new concept in terms of brainwashing. But there is some flow between adjacent capillaries that was unrecognized maybe 10 years ago. So when you change the pressure gradient, you're also changing flow in the parenchyma that could influence brain drug distribution.
Well, I mean, that's the point. When you put a drug into the ventricle, so cerebrospinal fluid is about a pint a day produced, and half of it comes from the choroid plexus, and the other half comes from the parenchymal fluid. And so when you inject methotrexate or any other is from the brain into the cerebrospinal fluid. So I'm just wondering, I mean, it's kind of a silly kind of question perhaps, but I mean, it's like swimming upstream. I mean, are we delivering, are we trying to deliver drug against a hydrostatic pressure? And that's because it's limited by the skull. I mean, now if the fontanelles are open in a mouse, I don't know much about mice and fontanelles, but maybe it's not an issue, but maybe but anyway, that's my question. And since this is an engineering institute as well as biological one, I wondered if anyone here might be interested in doing that.
Well, so some of the work, sorry, Bill.
Just quickly, I'm not an engineer, but I wish I was. I like engineers, but and this is one reason why gliadel didn't work, in spite of the fact that it was kind of developed here. But when the BCNU diffuses, it's really not related to flow. It's related to the sink effect, and we have as many capillaries as we have neurons. So we have a lot of capillaries. So it's a sink effect. You don't go very far before a highly permeable drug will get washed away. And when I say washed away, it leaves the brain by getting into the blood. So the flow rates are important, but this kind of concentration distance profile is really kind of dependent on this sink effect in the capillaries.
Yeah, so along this line, so the gliadel was a very exciting idea, and so we started some work with a colleague in the UK who has more of an engineering mind, where they've developed those self-assembly gels that now he can basically match the pressure into the gel to match the pressure into the patient's tumor. So during surgery, during the surgical resection, they can take a measurement of the particular pressure in that patient, match a self-assembly gel, and it allows the drug to distribute over like centimeters for days. So these are things that we're testing now with their group, but again, that's a very more simple approach of pressure. So we're taking a lot of things into consideration to get those drugs there.
Yes, excuse me, yes, thank you. So I was intrigued by the way you can quantitate the levels of the drug in your sections. Could you also determine the proportion of that active metabolite that was, even though in small quantities, you said it was distributed better than the actual drug, if I'm correct in understanding what you said? Can you also quantitate that as well?
So we could, so right now I just showed some relative intensity, assuming that they would have similar desorption ionization efficiency. It seems like there's a 10-fold ratio between the two, but some other works that we're doing intraoperatively where we follow metabolites, we use just isotopically labeled metabolites, and we can, we've done a study where we can mix this into the spray of solvent and matrix that we put on the tissue, so we can do, a calibration in an internal standard using an isotopically labeled metabolite. We don't have it, and we're not there yet. We haven't wondered about that, but yes, we can.
So I just had one quick question for you about SRS versus MALDI mass spec. And so with the SRS, the stimulated Raman scattering, you get really nice resolution. Beyond what you can do with MALDI mass spec. What prevents you from using the stimulated Raman scattering to actually look at the metabolites by looking at the different chemical stretches that are there in the metabolites that might not be there in the normal brain?
So we can, some of them, so it's always a debate almost every single lab meeting in my group where there's the mass spec people sending, debating with the SRS group here. But in fact, Elizabeth has done both mass spec and SRS, and she was able to image Erlotinib using SRS. So it just depends on some of those chemical properties of the compounds. But what we are seeing, so the pros and cons between the two is that SRS brings the higher spatial resolution, but does not have the sensitivity or the specificity that we have. So in those animals, we have not been able to detect Erlotinib yet. It was not high enough concentrations to do that.
Okay,
So
cool.